Hey, good morning, everyone. I introduced myself yesterday. I'm a nematologist, so I'm here to tell you what the nematodes are saying. Um, Nematodes are absolutely ubiquitous. This is a short little introduction. 7 out of 10 animals are nematodes. They saturate the trophic levels. They're literally everywhere, and they're still alive after being unthawed in 40,000-year-old Siberian ice, able to eat, reproduce. They're just absolutely incredible numbers of these animals. And they're actually great indicators of soil health as well. So they're a great metric for climate change. Specifically, plant parasite nematodes are of incredible agroeconomic importance. We have this number, $100 billion annually globally. This number is not from any real study. This is basically a survey of other nematologists and asking how much do we think nematodes cost and crop loss every year. And I think that underscores the importance of kind of this unknown metric from nematodes. We don't know how much damage they're causing. We don't know how much yield loss they're causing. They're often unseen underground. Their impacts aren't often as drastic as what we're seeing in this picture, with these tomato fields being very stunted and chlorosis and necrosis happening. There can be nematode infestations reducing yields by 5–10% without any above ground symptoms. We don't really know the impact of this. Moving on to climate change, I teach an introductory plant path course at UW Madison. And so we talk about the plant disease triangle quite a bit. And I just kind of want to highlight this idea that climate change impacts all nodes of this disease triangle. I'll be talking about two specific areas: high atmospheric CO2 and higher temperatures. Of course, those are two very closely linked. But hitting all nodes of the disease triangle, yes, you need a conducive environment, but higher temperatures, higher CO2, these have direct impacts on host. They have direct impacts on the pathogen, even when the environment stays conductive through these changes. So I just want to highlight that as well. So with insects, above ground pests, we see an increase in CO2. There's a greater foliar consumption rates. This is a really cool phenomenon, I think. As you get this higher carbon nitrogen ratio, pests need to feed more on these plants to get that nutritional requirement. And we see it in humans as well. As we talk a little bit about rice today, and there's this really cool project, the C4 rice project. If you can go Google that, I think it's really cool. Basically trying to change a C3 plant into a C4 plant to combat CO2 rates, as C4 plants are not susceptible to higher CO2. So you do get an increase in development time, a decrease in pest abundance. So this may seem beneficial in the long run. I'm not really quite sure, but initially the greater consumption rate is an issue. So how does this impact, this increased CO2 impact below ground sedentary pests like this root knot nematode life cycle we have here, where these migratory juveniles penetrate roots, and then they live the rest of their life inside the root, migrating towards the vascular tissue, forming these really neat giant cells, feeding sites. They become sedentary, they lose their vermiform structure, they actually lose all their muscles, and they become these big pear-shaped egg factories, essentially, where those eggs then hatch out from these egg sacs on these knots and reinfect the same plant over about a 30-day period. Again, it's very dependent on degree days as well. So how does that change? These aren't able to move to new sources. Once they've set up a feeding site, the nematodes are there. And so we set up this little experiment to look at exactly at this. We have two different tomato hosts here. The near-isogenic lines, one carries a nematode resistance gene and one without. Those are pretty much the only genetic differences between these two plants. Both infected with root knot nematode, a nematode that in fact can break this resistance. So it's going to be able to reproduce on both of these. It is under two different CO2 concentrations, 417, which is really close to what we have now, and 755, kind of the median what we expect in 50 years. And we see this thing where gall rating, kind of our rating of how much disease these root knot nematodes are causing, actually decreases, but only in one genotype under this higher CO2 pressure. So we're not seeing that increased feeding that we would see with above ground insect pests under high CO2. And I think that's really interesting to look at. But not all plant parasite nematodes are the same. There's different feeding behaviors, which are likely to respond differently to environmental changes. We have migratory endoparasitic nematodes, ectoparasitic nematodes, and then the sedentary nematodes as well. We've also, as someone did mention yesterday, there's also foliar nematodes as well. So it's just absolutely huge diversity. Not every nematode is the same, so I can't cover them all today, unfortunately. But going back to not all nematodes are the same, we're going to take just one species of nematode, not nematode, one guy named Hapla specifically, and we have two different isolates, one from France and one from California, so two very distinct geographical locations. They have massive different phenotypes and behaviors, and also they induce different pathways in plants. So here we have this orange nematode from France is increased in expression of a plant gene, while the other one actually has a decrease in that same plant gene expression. So just to show as a concept, what we can do is across these nematodes, they can mate, we can create recombinant red lines, and then we can essentially map down what loci in the nematode genome are responsible for the increase in plant gene expression. So we can do this across all the plant genes simultaneously, and across all the nematode genes simultaneously as well. Make these massive genetic maps, which are really quite cool. And we get these nodes and these genetic interacting pathways of plant genes that seem to be and nematode genes that are responsible for this interaction that's different between these two different isolates. So as we look at nematodes or any pathogen moving globally with climate change, it's important to know that not every pathogen is the same. Not every genotype is going to be the same and respond differently. But we do see this across multiple different hosts. So when we have these two different lines of nematode, LM and BW9, we see this difference in gene expression. We can go to different hosts as well, such as tomato and soybean, and we can see that same gene, just homologous genes in these different hosts acting similarly to these two different pathogens. So again, I just kind of want to underscore that not all pathogens are the same, not all nematodes are the same. And so I showed you root knot nematode, a sedentary endoparasitic nematode, how its impact on high CO2 was genotype driven, but also had seen a lower disease metric. Other nematodes act more herbivorous, act more like a foliar insect, so those kind of moving through, migrating, eating on plant tissue. This is one, Protolenchus penetrans, a root lesion nematode, and this one we would expect to have very similar interactions with CO2 that we would see with our above ground insect pests. And so this is something that our lab is now looking into and trying to understand these interactions between climate change factors and different nematode feeding behaviors. That was kind of high CO2, but if I want to move on now into higher temperatures, we know, yes, global temperatures are increasing, but what I think is really interesting is that your nighttime low is actually increasing faster than your daytime high. And what happens then is the difference between your daytime high and your nighttime low is actually decreasing. That's a very important cue for plants. And we think it's going to be a very important cue for pests that feed on plants that have these intimate interactions, like root knot nematode, forming these feeding sites and interacting with these plants directly. So we have here just two different lines of tomato, a wild and a cultivated tomato. And we have them under two different regimes, a warmer nighttime temperature and a normal nighttime temperature. The difference about 4 degrees Celsius at night. We have here just one gene where we don't see a difference in a warmer nighttime temperature between gene expression between these two different lines of tomato, but we do see a difference in a normal nighttime temperature. And we can see the opposite be true for a different gene as well. And so plants are responding differently to these different temperature regimes. Every gene is going to act slightly differently. And with that, we can also see that nematodes infected on these different lines will have different gene expression under these two different temperature regimes as well. And so right now we have a funded project through USDA and NSF to kind of look through the plant genome. Now I showed you nematode kind of recombinant red lines. Now we're taking tomato recombinant red lines and doing the same kind of thing. Where we have 130 recombinant red lines, inoculating which is one strain of root knot nematode, looking at gene expression, looking at gross plant morphometrics and phenotypes, and getting down to what are the genetic loci responsible to interact with all these abiotic and biotic stress. So the plant is dealing with this biotic stress of the nematode and also the abiotic stress of the higher or warmer nighttime temperature. So what are those responses in the plant and how do they deal with both these happening simultaneously? So we have these 4 different conditions, either warmer nighttime temperatures and infected or normal, not infected, and so on. Just the very classic 2x2 grid there. This is going to produce another gene interaction network for all of this, understand what are these keystone genes in this interaction that kind of regulate plant responses. And as I've shown you before, we've seen these genes being able to have the similar responses across this wide nematode host range. So we're expecting this to be just a model that we can use to understand how many plants will interact to abiotic and biotic stresses. So I just have a couple of preliminary graphs. We're still in the middle of this project. And so we can look at, the CO2 assimilation of the leaves under these different conditions with normal nighttime being inoculated or uninoculated. We can start seeing some differences there. I didn't put any stats on this because we don't have all of the data points in yet, and we haven't done it in the RNA-seq yet either. We also look at infection rate, a transpiration rate of plants. So we're just taking in an absolutely incredible amount of data on how plants are responding to both of these stresses simultaneously. So that's what I wanted to talk about in terms of climate, but I did mention briefly in the beginning that we don't really understand where nematodes are. We don't understand what diseases they're causing, and so there's really a critical need for centralized nematode identification tools. We're losing a lot of that. There's a national surveillance of emerging, we need to increase national surveillance of emerging nematode threats. We really don't have the tools in place for that. Diagnostics are very laborious on nematodes currently. We're losing the specializations to be able to identify nematodes right now. We're not really collaborating nationally on a national level to watch nematode movements, or even state to state. There's very different tools that people are using to identify what nematodes they have in their fields. So we need to update these, and so we're working on that. One example is this water-ridden nematode in Woodbine Antilobii. Right now, we're saying it's only found in North Carolina, but that's absolutely not true. There's a whole USDA-funded program called Find Me. This nematode is moving all throughout the Southeast, but it's not being reported on a national scale yet. We're not able to find it. This nematode was found actually in South Carolina because a nematologist took a job at USDA and went to the grocery store and found it on sweet potato. And so that's how we're getting our information of nematode distribution in the US. We need to do a better job. So it's been there. It's likely been there for some time, but because there wasn't a specialist that would recognize it in place, we're not actually capturing it. And so we need tools available to people to identify nematodes more remotely, that we can do it on a much larger scale and a much more high throughput mode. So currently it's nematode extraction, it's microscopy, it's reporting, it's very laborious. Here's a sample of nematodes that you can get out of just a handful of soil. It's a massive number of nematodes. Not all of these are plant parasitic. So you have to go through each one of these, find which ones are plant parasitic, and then identify them to genus or species level. Often it's too numerous to count. There's often sample fatigue and sample bias. Different nematologists will count different numbers of nematodes, and again, that specialization that they can recognize Persephalancus versus Modigina versus Persephalancus versus so on and so on. That specialization becomes really a bias factor as we try to understand what nematodes are out there in the field. I actually want to consider, we're trying to find new pests coming into areas. We need someone that doesn't just recognize, okay, this is a corn sample, I'm expecting to find XYZ, I'll go look for those nematodes. We need to look for things that might not have been there previously. And so that's where that sample bias becomes really quite important. So to do this, we're developing machine learning algorithms, basically. It's just base ID for nematodes. We created large data sets from nematode assay labs, samples that come in. Had to hand annotate a lot of these samples, looking for stylettes, the B marker for plant parasitic nematodes, and annotate these down to the genus and species level as well for each individual one. There's so many different important nematodes. We're trying to focus on the ones that are most important, the ones that we think can move greater distances. But that's what we're looking at right now. So there's a lot of problems with this. There's a lot of issues. There's no current large image database that exists for nematodes. You search nematodes, on Google image search, and not many images come up. And so we have to create all of this. We have to generate this, and of course, with AI, more data is better, and so we constantly have to keep annotating images. But we can do pretty well. We can get just, if it's plant parasitic, we can get 80% right, which is really quite good. That's about as accurate as a nematologist would look at this image and be able to detect, because again, it's so many nematodes in such a small sample size. I'll have so many samples, massive batches, 7,000, 30,000 samples a year. You're going through a lot of samples, and so you're not going to catch them all as a human either. And 80% usually is pretty good. The thresholds are often quite large. So if you have 200, you don't have a problem. But if you have 2,000, you've got a problem. And so that's what we're trying to find. So we can get developing this all in one tool. We put in a grant for the USDA Biosecurity Panel. We're hoping to hear from that soon. That's where we're at with nematodes. We talking about some of the take-home messages. I wanted to highlight again that climate impacts all nodes of plant disease triangle. Nematodes are incredibly diverse. Impacts of climate are likely to have very different outcomes for different feeding habits of different nematodes, even on different genotypes. So my current research is focusing on isolating these kind of genetic keystones for responding to the abiotic and biotic stress. And nematode diagnostics do not currently capture the importance of the impact of nematode disease. We're trying to work towards improving that. Any questions? Jim.
Yeah. Microphone. I've been in too many meetings. Oh, that's really interesting. So are there interactions with nematodes with other pathogens that make a complex disease situation? And that's something that we've never really touched on.
Yes.
And then
so
secondly, the microbiomes and their the good and bad microbiomes and their interactions with nematodes would be huge as well.
100%. So definitely root lesion nematode can be examples for both of those. Potato early dying, there's a nice synergistic interaction with verticillium and that. And we know if you have low nitrogen, which is basically increasing your carbon nitrogen ratio in plants, you see more of that happening. And so that might be because the nematode is not creating more lesions because it's migratory, it's feeding like an insect would on a high CN plant. And that gives more routes for verticillium. So yes, we are already seeing that. But certainly the microbiome crop rotations with soybean cyst nematodes and the microbiome is very important in delivering kind of the fecundity of that nematode. Feeding with Palinkas again, there are some endosymbiotic bacteria that seem to be important in those interactions and in feeding. So yes, if there's too many factors, I would say.
Yeah.
There's complex on strawberries too.
So Jan kind of went where I wanted to go, and that's about what do you see or can you tell at this point if there is any impact on the diversity of the microbial population in general. So think about some of the potential nematode trapping fungi, they end up being impacted so that you're seeing populations decline. Are you seeing decrease in the diversity of the nematodes in soil profiles?
So I
don't have a background to even know that.
I don't have enough background to know that. We haven't gone that deep in the microbiome. We'll look at nematode populations. They're a good indicator of soil health. The rest of the microbiome becomes really massive, right? We've done studies where you just move a foot over in the soil and all of a sudden it's completely different. And so we don't have the numbers yet. I don't think we have enough data for that. I'm looking for AI to help us out with that once we get enough data sets to come in. But right now, the return on making those data sets isn't high enough because you can't get a paper published just showing differences between different sites, right? That's not enough. So.
But it really speaks to the importance of having this baseline data, so we can have some idea what really is happening. Because there's just not enough information. Jim?
I don't see many reports on how often or how much we're moving nematodes around in the plant trade. We get lots of numbers on fungi, bacteria, viruses, but I don't really see much on the nematode side.
No one's looking. The tools are not at that level where you can look at it that quickly, right? I mean, we need the AI kind of tool to be able to do that, where we can just take an image really quick. But even the extraction, the microscopy, it's so much time and energy devoted to this one image, and you can't just sample one thing. You need to be sampling, a lot of different samples and across everything to try and capture it. So it's really needle in the haystack issue. I don't know how that's going to be solved. Besides getting sampled from the field and saying, now we've got it, and not getting it as quickly as possible.
I think it'll be more comfortable. Soil fungus is a whole category system. So we're importing soil all the time from all over the planet, and we're not looking. Yeah, this company.
This is Allison. I'll just say, so my number one crop coming into my lab is garlic. And it's because we do garlic seed testing for Didylungus dipsaki nematode, and it's a binary test in our lab. Like, is it present or is it not? And I have to look at every single sample because I don't trust a student to do it. And it would be, and one thing you didn't mention is that there's hardly any PCRs out there for nematode testing. And our lab is trying to design one for it so that I don't have to be in the lab all the time looking for these and not wasting, but using a lot of my time, which then
Very
highlights the
difficult-
AI.
Very difficult to do a PCR test for nematodes without
Yeah.
worrying about false positives, especially something so sensitive where if you just have one, you're in trouble. A PCR will need a lot of false positives, and it might not be that is the major concern.
But false positive would be fine. I could
Okay.
then do it manually. False negative is where it'd be concerned.
Okay.
But anyways, it underscores the AI piece as well as we need good diagnostics to know what the heck is happening. So,
You didn't touch on entomopathogenic nematodes. I just wondered what you have any comments on that?
I think they're wonderful. I did try a study when I was in Florida, looking at how different EPNs can persist in different climates, but COVID hit, we were able to had to end up scrapping that project. So we're looking at persistence, more from a biocontrol. Can we get EPNs to persist in different environments? How can we basically bioengineer an EPN with the right symbiont to be able to persist in different environments or sustain environments that are changing?